Welcome to G Fun Facts Online. Imagine a butterfly, a flash of yellow and black against a summer sky, its wings carrying the delicate weight of its own survival. Now imagine that butterfly's future, its entire genetic legacy, resting within a tiny frozen egg, suspended in time at a temperature colder than an Antarctic winter. This isn't science fiction. This is a real high-stakes scientific mission, a dance with ice, that could save one of the world's most iconic insects, the swallowtail butterfly. For one particular subspecies, the British swallowtail, the music is fading fast. Confined to the unique marshy fenlands of the Norfolk Broads in England, this beautiful insect is living on a knife's edge. Its world is shrinking, its food is disappearing, its population has plummeted by an alarming 57% in the last 2 decades, and 2024 was its worst year on record. As traditional conservation methods struggle to keep up, a team of scientists is turning to a radical new solution, a frozen ark. They are embarking on a pioneering journey to cryopreserve swallowtail eggs, creating a genetic safety net that could pull this butterfly back from the brink of extinction. Today, we're going to explore this fragile majesty, dive deep into the mind-bending science of freezing life, and ask whether this audacious endeavor could revolutionize how we protect the small creatures that run our world. The family of butterflies known as Papilionidae, the swallowtails, includes over 580 species worldwide. They are the giants and jewels of the butterfly world, but their beauty makes them vulnerable. More than 60 species of swallowtails and their cousins, the birdwings, are in decline, threatened with extinction. The story is a familiar one in our modern age. Deforestation, the draining of wetlands for agriculture, pollution, and even the illegal trade of rare specimens. The British swallowtail, a unique subspecies called Papilio britannicus, is a perfect, tragic case study. Its entire existence is tied to one specific habitat, the fens, and one specific plant. Its caterpillars will only eat milk parsley, a feathery plant that thrives in these wetlands. No milk parsley, no swallowtails. It's that simple. And these vital habitats are themselves under an existential threat. Rising sea levels, a direct consequence of climate change, are predicted to push saltwater into the freshwater fens. This process, called salinization, would destroy the milk parsley, effectively starving the British swallowtail out of its last remaining stronghold. With its population already classified as vulnerable on the Great Britain Red List, the situation has spurred conservationists to think beyond planting more food and managing land. They need an insurance policy, a backup plan, and that's where the idea of a frozen ark, a biobank of cryopreserved genetic material, has become a vital glimmer of hope. In a trailblazing project, a powerful alliance has formed. Researchers from Anglia Ruskin University have teamed up with the conservation charity Nature's Safe and Jimmy's Farm and Wildlife Park, a place famous for its conservation breeding. Their shared mission is to determine if cryopreservation can be a real, viable tool for saving the British swallowtail. Now, given how incredibly rare the British subspecies is, you can't just go out and start experimenting on their precious few eggs. So the research is initially being conducted on a close, more abundant relative, the European swallowtail. A thriving captive population of these butterflies at Jimmy's farm provides the perfect subjects for this crucial, foundational research. The process is both simple in concept and incredibly difficult in practice. Researchers will collect freshly laid eggs from the European swallowtails. These tiny eggs will then be plunged into liquid nitrogen, reaching a staggering temperature of -196 degrees Celsius. At that extreme cold, all biological and metabolic activity virtually grinds to a halt. The embryos inside are placed in a state of suspended animation, frozen in time. The real test, of course, comes later. The scientists will attempt to thaw the eggs and rear butterflies from them. They will then meticulously compare the health, development, and, most importantly, the reproductive success of these cryopreserved butterflies to a control group that hatched from normal, unfrozen eggs. Dr. Alvin Heldon of Anglia Ruskin University highlighted just how new this is, stating, "Cryopreservation is a promising tool for supporting conservation efforts, but we believe this is the first time it has been attempted with butterflies. If this works, it's a game changer. The method could be used to store the eggs of the precious British swallowtail, creating a genetic reservoir that can support long-term conservation for decades or even centuries to come." So how exactly do you freeze life without destroying it? This is where we get into the truly fascinating science, a delicate dance between physics and biology. Freezing a living organism isn't like making ice cubes. The biggest enemy is the ice itself. When water freezes slowly, it forms jagged ice crystals. Inside a living cell, these crystals act like microscopic shards of glass, puncturing membranes and destroying the delicate internal machinery. The cell is literally shredded from the inside out. To get around this, scientists don't try to freeze the cells, they try to vitrify them. Vitrification is a wild concept. It means turning to glass. Instead of letting water molecules arrange themselves into the orderly destructive crystal lattice of ice, the process rapidly cools the water into a disordered, solid, glass-like state. It's solid, but it has the molecular structure of a liquid, with no sharp crystals to cause damage. To achieve this, scientists use chemicals called cryoprotective agents, or CPAs. Think of them as a kind of biological antifreeze. Chemicals like ethylene glycol and a sugar called trehalose are introduced to the embryos. They perform a crucial two-part job. First, they draw water out of the cells, dehydrating them. Less water means less potential for ice to form. Second, they interfere with the water molecules that remain, preventing them from linking up into crystals during the rapid cooling phase. But this process is a tightrope walk. The cryoprotectants themselves can be toxic. The embryos must be exposed to them for just the right amount of time, at just the right concentration, to achieve dehydration without being poisoned. It's a chemical balancing act with the life of an organism at stake. And with insect eggs, there's another major hurdle. Insect eggs have evolved over millions of years to be remarkably tough. Their outer shell, called the chorion, and a waxy layer just beneath it, are designed to prevent water loss and protect the embryo from the environment. This is great for surviving in the wild, but it's a huge problem for cryopreservation, because these protective barriers also block the entry of the vital cryoprotectant chemicals. A key part of the Swallowtail project, therefore, is finding a way to gently poke holes in this armor. Researchers will have to experiment with methods to permeabilize the eggshell, perhaps using chemical treatments like a very mild bleach solution or alkanes to temporarily break down the waxy layer, allowing the CPAs to get inside where they're needed. Timing is also everything. There is a Goldilocks window in the embryo's development. If you try to freeze it too early, it's too fragile. If you wait too long, it might have already developed complex structures or internal barriers that are impermeable to the cryoprotectants. Finding this perfect window is painstaking work. For the sunflower moth, a species that has been successfully cryopreserved, scientists found the optimal stage was between 45 and 55 hours of development at a constant 21 degrees Celsius. Similar meticulous research will be needed to find the perfect moment for the swallowtail. Finally, after all that, there's the thaw. Getting the egg back from -196 degrees to room temperature is just as perilous as the journey down. If you warm it too slowly, a process called devitrification can occur. The glassy water can suddenly snap back into its crystalline ice form as it warms up, causing the very damage you tried so hard to avoid. To combat this, scientists are developing incredible ultra-rapid warming techniques. Some labs are even using powerful infrared lasers that can warm samples at unbelievable rates, some as high as 10 million degrees Celsius per minute. This blasts the embryo through the danger zone so fast that ice has no time to form. The ultimate measure of success isn't just seeing a caterpillar hatch. The team will need to confirm that these butterflies grow into healthy adults, that they can fly, feed, and most critically, that they can reproduce successfully, passing on their genes to the next generation. Only then can the genetic arc be considered truly viable. Now, while this is a first for butterflies, the cryopreservation of insects is not entirely uncharted territory. This provides a foundation for hope. The field really began back in 1990 with the first successful cryopreservation of fruit fly embryos, a landmark achievement that proved it was possible. Since then, protocols have been developed for several other species. For instance, researchers working with the sunflower moth achieved a 23% hatch rate from their vitrified embryos, and of those, a solid 60% survived to become healthy adults. In another remarkable study, embryos of the green blowfly were successfully cryopreserved, stored for 8 years, and then thawed. Over 8% of them hatched and developed into perfectly normal flies. These numbers might not sound huge, but in the world of conservation, they are monumental. They prove that the formidable biological and physical challenges can be overcome. The knowledge gained from these earlier studies, on everything from membrane permeability to cryoprotectant toxicity, provides an invaluable template for the scientists working on the Swallowtail project. They aren't starting from scratch. They are standing on the shoulders of giants in the microworld of insect cryopreservation. And the implications of this research extend far beyond the beautiful swallowtail. If a reliable protocol for freezing and thawing butterfly eggs can be established, it could completely revolutionize insect conservation. The technology could be adapted for other endangered butterflies and moths, and potentially for a vast range of other threatened insects. Insects are the unsung and often unloved heroes of our ecosystems. They pollinate a huge portion of our crops, they break down waste, they aerate the soil, and they form the base of countless food webs that support birds, fish, and mammals. Yet they are facing their own quiet apocalypse, an extinction crisis that is often overlooked in the shadow of more charismatic animals, like pandas and tigers. Keeping endangered species alive in captivity is one option, but it's incredibly expensive and logistically complex. Furthermore, over generations, captive populations can suffer from genetic drift and inbreeding, losing the genetic diversity and fitness that made them successful in the wild. A cryopreserved biobank, or a frozen ark, offers a powerful solution. It's a scalable and relatively cost-effective way to safeguard the genetic diversity of thousands of these vital creatures. A frozen backup of the original genetic stock from the wild could be used to refresh and revitalize captive populations, ensuring their long-term health and giving them a fighting chance if reintroduction becomes possible. Of course, the flight path ahead for the Swallowtail project is uncertain. There is no guarantee of success. The intricate dance of variables, the precise chemical cocktail, the exact developmental stage, the speed of cooling and warming, will require years of painstaking research, trial, and error. But in the face of mounting threats and the accelerating loss of biodiversity across our planet, timid steps are not enough. Bold, innovative, and even audacious solutions are not just desirable, they are essential. The project to freeze swallowtail eggs represents a powerful beacon of hope. It's a testament to human ingenuity and our capacity to find scientific solutions for the problems we've helped create. It's a proactive step, a way of preserving the very essence of a species before it vanishes forever from our world. The fate of the iconic swallowtail may well rest in a frozen embrace, a dance with ice that could secure its future for generations to come. And in doing so, it may just pave the way for a new era in the conservation of the small, often overlooked creatures that are the true engines of our planet.